I guess today we'll talk about immune mechanisms. In that, we'll talk about what is immunity and how it is classified and what is cell mediated immunity. And in another video, I'll make what is humoral immunity. So, immunity is nothing but basically a defense against the microorganism. So, immunity can be by birth, we call that as innate immunity, and it can be acquired in the process of aging or in the lifetime, it can be acquired. So, innate is inborn and is dependent on genetic. And acquired immunity, it is of two types, active and the passive. Active immunity in the sense our body is active and producing its antibodies. Whereas passive in the sense it is passively receiving the antibody. Coming to the active, like a natural infection, that is a natural active. Whereas artificial induction by vaccines, then passive, natural from maternal, either through placenta or from the breast milk, or artificial by directly giving the immunoglobulin like anti-D immunoglobulin. So, inborn immunity, and it is non-specific. Any organism, it acts with the same mechanism and it's not specific. And mechanisms are simply a barrier, like skin and mucosa. If the skin and mucosa are intact, they don't allow the microorganisms to go in. If there is a cut, there is an infection. There is a chance of entry. So, mechanical barrier. Surface secretions like saliva, tears, gastric juice, sebaceous glands, all will be having antimicrobial activity. Humoral defense mechanism like complement system, even the complement system can be activated by antigen antibody complex, it is not specific. And interferons, which are antiviral, and basic polypeptides like protamine and histone. And cellular mechanisms like phagocytes, like neutrophil, it's not specific, anything that comes in the way it eats up, macrophages. And natural killer cells and eosinophilic granules. All these are cellular mechanisms. All these comes under innate immunity. Coming to the acquired immunity, as we said, during his lifetime, it's called acquired immunity and it is specific. And it's active and passive. Active acquired immunity in that natural, so is due to subclinical or clinical infection. Infection is the reason here. And artificial active immunity here by introducing antigen into the body, either it may be a live attenuated or it may be a killed vaccine. So like a BCG and polio vaccine, this is artificial active immunity. And coming to the passive immunity, immunity due to transfer of ready-made antibodies. So the ready-made antibodies can be transferred by naturally, like through breast milk, that is immunoglobulin A, or through placenta, that is immunoglobulin G. And artificial passive immunity by injecting antibodies like anti-tetanus, anti-D antibodies. So natural is infection, artificial is vaccine. This is active. Passive is ready-made antibodies by maternal antibodies or by monoclonal antibodies. So, active immunity is actively produced by host and it receives passively by host, passivity. And there is a latent period. As they are ready-made antibodies, no latent period. And there is immunological memory, so there is the secondary response and there is no secondary response, no immunological memory. And it is long-lasting and it is short. And active immunity can be applicable to immunodeficient individuals. Even immunodeficient individuals can take passive immunity or passive antibody. And before going to the actual immunity, we should know what is an antigen and what is an antibody. So antigen is the one which stimulates immune response. And it may be a protein either combined with the glucose, glycoprotein or lipoprotein. And every antigen is specific because of a specific region called as epitope. We can see here the antigen is having a shape and the antibody is having a similar fitting shape. And similarly here. So this region is called antigen determinants. We call them as epitopes. And coming to the antibodies, they are produced by the plasma cells, which are nothing but activated B lymphocytes. And it is a Y-shaped structure made of 4 polypeptide chains, 2 heavy chains and 2 light chains. Heavy chain is around 50,000 Daltons molecular weight, and based on the heavy chain, the immunoglobulins are named. So IgA, alpha heavy chain, gamma heavy chain, IgG, delta heavy chain, IgD, and epsilon, IgE, and mu, IgM. So the immunoglobulins are named according to the heavy chains. And each immunoglobulin will be having an amino terminal and a carboxy terminal. And towards the amino terminal, we have the variable regions for both heavy chains as well as for the light chains. And these variable regions are specific for that antibody. And the carboxy terminal will be having the constant regions. So constant heavy chain 1, heavy chain 2, heavy chain 3. And the 2 heavy chains are joined by disulfide bond. Light chain will be 2 types, kappa and lambda, and it has 2 regions, variable light chain region and constant light chain region. And what are the functions of these immunoglobulins? So immunoglobulins protects IgG can cross placenta, and also it is the important immunoglobulin in the blood and body. IgA, we call this as secretory immunoglobulin, and it is a surface immunoglobulin. So secretory as well as surface. IgM, M for mega, and that is the larger immunoglobulin, and it protects the blood. IgE is responsible for type 1 hypersensitivity reaction, and the IgD activity is not clearly known. Now we'll talk about cell-mediated. Cell-mediated immunity is the immunity by T lymphocytes. So in the acquired immunity, we have 2 kinds, cell-mediated and humoral immunity. So we'll talk about cell-mediated immunity now. So cellular immunity, where it comes into action? 1. Fungi, viruses, and intracellular pathogens like Mycobacterium tuberculosis and Mycobacterium leprae. And it is also concerned with allograft rejection. So any transplant by the other fellow can be rejected by the cell-mediated immunity, and it is responsible for type 4 hypersensitivity. So type 4 hypersensitivity is the only one which is cell-mediated, remaining are all immunoglobulin mediated. And even the immunity against the cancer cells. And types of immune response, there are always 2 responses, primary and second. Primary immune response or primary cellular response is the one which occurs when the antigen enters first time. And then when it enters the second time, the response is called as secondary response. So for the cell-mediated immunity, first the antigen has to be processed by a antigen presenting cell. So we have 2 kinds of antigen presenting cells, macrophages and dendritic cells. These are the antigen presenting cells. What they do? They work along with MHC antigen, major histocompatibility complex antigen. So whatever the microorganism that entered, so like this is the microorganism that entered the antigen presenting cell, that is dendritic cell or macrophage. Now it breaks that and it combines with a major histocompatibility complex, and it presents it to the surface. So if this is the pathogen, sorry, this is the macrophage and this is the pathogen, and this pathogen is having a antigen determinant vaccine. Now a major histocompatibility complex is synthesized, and this pathogen specific part is attached with the pathogen. So this is now recognized by the T lymphocyte. So antigen presenting cell break this bacteria and presented it to MHC, and that was shifted to the surface receptor. Now based on what it is attached, if it is attached to MHC1, it will be recognized by CDH, or which is type T cytotoxic cell, cluster differentiating factor 8, which is concerned with the T cytotoxic. If at all, if it is attached to MHC2, it will be recognized by T helper cell or CD4 cell. So CDH cells will recognize MHC1 and CD4 cells will recognize MHC2. Now, the CDH cells will differentiate into T cytotoxic and T suppressor cells. CD4 cells will differentiate into T helper cells and delayed hypersensitivity cells. And some T lymphocytes transform into memory cells which will be responsible for the secondary response. Now, again the same. So the infected cell broken down the bacteria to the antigen fragments and the MHC is synthesized and both coupled together forms antigen MHC complex and that is presented on the surface, and as one MHC1 is presented, it will be recognized by the cluster differentiating factor 8 cell, that is cytotoxic T cell. Now, similarly, if it is present to the MHC2, it will be recognized by the T helper cell. Now, when the T cytotoxic cells are formed, they will kill the pathogen directly by producing perforates. So, which is nothing but they will punch holes on the membrane of the pathogen, which is a calcium dependent. And also they can induce apoptosis by secreting tumor necrosis factor B beta. This is also called as lymphotoxin, which kills the bacteria by increasing the calcium content. And also by secreting interferons, gamma interferons, which are antiviral. Now, this is the way of the activated T cytotoxic cells punching the holes in the pathogen attacked cell, and that will cause lysis. Now, T helper cells, what they will do? These T helper cells will secrete interleukin 2, interferons, and tumor necrosis factor beta. And this interleukin factor 2 will cause furthermore synthesis of T cytotoxic cells or T suppressor cells. And T helper cell 2 will secrete interleukin 4, 5, 6, 10, 13, which all will cause activation of B lymphocytes. So T lymphocyte activation will also cause the activation of B lymphocytes. That is called TB cooperation. Now the T suppressor cells, what they will do? So they will suppress the activity of cytotoxic and helper cells, and these cytotoxic cells are prevented from destroying the cell tissue. To summarize this, we can say that bacteria entered the antigen presenting cell. Now it is engulfed and is broken down, and that is presented on an MHC, and this is recognized by a T helper or T cytotoxic depending upon MHC2 for T helper, MHC1 for T cytotoxic. Now the cytotoxic and also this T helper differentiate again to will also produce T suppressor. This T suppressor will check the T helper and also T cytotoxic from overacting. So, antigen is processed, present to MHC, and that will cause activation of T lymphocytes that will secrete lymphokines, which will cause further proliferation of cytotoxic and suppressor cells, and also activate T lymphocytes, which produces immunoglobulins. Thank you for your patient listening.